This video is going to get us through the digestive system in lab. This lab starts on page 175 and it's lab number 14. First off, they give you some instructional objectives, but basically we're going to be learning about the anatomy and physiology of the digestive system. After that, it talks about some of the processes that are necessary in order for you to digest your food. And the lab manual goes with, there's 5 steps. Some books say 6 steps, but I'll mention the 6th one here in just a second. And so you can always pause the video to get it, but your lab manual already says this. So the stuff that I'm going to hit. The step that is missing from this is propulsion, and propulsion is what pushes the food through the digestive system, and of course that's a very important process. There are a lot of people who have slow propulsive systems in their digestive system, and that tends to make them more likely to develop constipation, which has complications that lead up to colon cancer. And so that propulsion is important, even though it's not mentioned in the lab manual. The other thing that I'll mention is I've not ever heard anybody else use the term egestion, and so I am not going to use it. Most textbooks just say elimination or defecation, and so those are the terms that I'm used to using. But at the end, there are nutrients that you can't actually absorb from the food that you eat, and those things have to be eliminated, and so they just pass right through you, essentially. All right, so this is a model that we would have studied had we been in a face-to-face class. So that's why I'm going to show you this picture and kind of run you through the basic anatomy as we go through here. You already have a labeled version of this picture on page 177, but just to kind of hit you through what each organ does. So, mouth. I mentioned ingestion on a previous slide. Ingestion is the process of eating, so that's putting the food in your mouth. And so you guys all know where your mouth is since that's where you all put your food. There's also a lot of mechanical digestion that happens in the mouth. So mechanical digestion is when you physically tear your food apart, and teeth are really good at that. They're not the only things that do that, but they're a big chunk of it. There is a little bit of chemical digestion. There are some enzymes in the saliva that you produce into the mouth, and that's really just a slight amount because your food doesn't spend enough time in the mouth to really be digested all that well. Once you swallow food, it's going to enter into the pharynx, which we talked about a little bit in the last chapter, the respiratory system. Pharynx is also the throat, so it's really part of 2 systems, the respiratory system and the digestive. At the bottom of the pharynx, that's where you have that switching point where your respiratory system separates and becomes the larynx and then the trachea. And then towards the back, the pharynx is going to be continuous with the esophagus, and you can see that continuation going on right there in that model. The esophagus is the tube that takes the food from the pharynx, which is going to be stopping right about there, down to the stomach. So from here to here, all of that's the esophagus. And once again, food's not in there long enough to really do anything with it, so it's really just a passageway for food to get from point A to point B. Stomach. So all of this right there is the stomach. There are lots of little parts. They went ahead and put those in parentheses there, but you're not A&P students, so you don't care about most of those. The stomach has a bunch of folds in it that makes it a very expandable organ. You can eat a lot of food and not tear the stomach because those folds do help to make it very expandable. You produce a lot of acid in your stomach. That acid helps you break down the extracellular matrix between the cells, and it helps you break down some between plant cells as well, even though they don't have a matrix. Let's see, there's also an enzyme called pepsin that gets secreted by the stomach, and that helps you digest the proteins that are in the food that you eat. So there's really a lot going on in the stomach. Food can spend a fair amount of time in the stomach. It kind of depends on what kind of food you're eating. Like if it's a soup, it's going to pass through fairly quickly. But if it's something that's a little bit more complex and more difficult to digest and liquefy, it's going to spend some more time in the stomach. And if you ate a very large meal, it's going to take time for the stomach to empty after that as well. There's a little door at the end of the stomach. Go away, little window. That's right here. That is the pyloric sphincter, or what this lab manual is calling a pyloric valve. That helps control when the food does get to leave the stomach and go to the small intestine, because again, things have to be liquefied enough that we want to pass it on. Otherwise, it needs to stay in the stomach until we have it digested down enough. The liver does so much in relation to digestion, and it has functions that play a role in the immune system. It's a very important organ. As related to digestion, it produces something called bile salts. Those bile salts are then going to be sent to the gallbladder, and the gallbladder is going to store the bile and release it after you eat a fatty meal. One of the other things that the liver does as related to digestion is, should you consume something that is perceived as toxic by your body, like alcohol, your liver has to detoxify that. And so that's what it means by detoxifies chemicals in the bloodstream. And of course, you ingested alcohol. That's how it got into your body. So that relates back to this system. Gallbladder, I already mentioned earlier, but again, it stores bile and releases it after you eat a fatty meal. Pancreas sort of hides in this picture. I've always thought it looks kind of like a tongue. It's this little structure right here with that white line that runs down the middle of it. The pancreas is another one of those organs that belongs in a couple of organ systems. It belongs to the endocrine system because it makes some hormones that are necessary for regulating blood sugar levels, like insulin. But it's also part of the digestive system because it makes a bunch of enzymes that help you digest every single major category of food. And so the pancreas is critically important for digestion. Small intestine has several different chunks or sections to it. You have the duodenum, which is just the first foot. Despite the fact that it is the smallest segment of the small intestine, though, both the liver and the pancreas release their products into the duodenum. There was actually a little hole right there in the model. So that means a bunch of chemicals are actually first starting to mix in with your food there in the duodenum of the small intestine. The next chunk, which is somewhere around 8-ish feet, it depends on each person, but during the jejunum, that middle section, that's where we're doing most of the chemical digestion and breaking the food down into small enough molecules that you can actually absorb it into the body. And then the ileum is the last about 10-ish feet of the small intestine, and by the time we've made it there, the food molecules are small enough that that's where most of our absorption is going to happen. Now, the small intestine as a whole, the A&P textbook that we use says that it carries out 90% of your nutrient absorption. So that means the stomach isn't helping you absorb your nutrients, it's helping you break it down. Same for your mouth, and your large intestine is not absorbing it either. It's the small intestine that does almost all of that. So it's responsible for getting the nutrients out of your food and into your body. The ileocecal valve is the little sphincter between the ileum of the small intestine and the large intestine. It's not a very good sphincter, but it does exist. The cecum is this one little pouch right here that hangs below the ileocecal valve, which was right there. It's sort of a blind-ended pouch. In some animals, the cecum gets to be very long. Those are what are known as hindgut fermenters. We are not that, and so our cecum is super tiny. Hanging off the bottom of that, you got this little guy right here, which some people think looks like a worm, so it is called the vermiform appendix, or wormy appendix. Your lab manual just calls it the appendix, and that's fine. Here's what kills me. So they put vestigial structure. Vestigial means it has no function, but here's the thing, there is a function. It plays a huge role in your immune system, and so you can't say it's a vestigial structure, so I'm just going to cross that out. I didn't make this slide, but the appendix does have a job, and your immune system does get weakened slightly by taking the appendix out. However, if they're taking your appendix out, it was going to kill you, so it needed to come out. All right. Colon is also known as the large intestine. It's all of this gray stuff. This is actually part of it. This section right here was supposed to go right there. Then we've got this little chunk. That's all colon right there. This absorbs water out of what, by the time we make it to this point, is starting to resemble feces, although it's very, very runny at the beginning of the colon, and it doesn't start to look like what you're actually, I'll just say, pooping out, for lack of a better term, until you get towards the end of the tube, and that's when it actually starts to look like whatever hit your toilet when you get there. The rectum is this nice, it looks like a little water slide, a horrible, horrible water slide. It just stores feces so that you can release it at a later time. And then the anus is the sphincter that controls when you release that feces from the rectum. All right, after that, page 180 starts to talk about the chemical breakdown of food. So a thing to know, anytime you see a word that ends in ASE, that means it's an enzyme, and usually what comes before that ASE tells you exactly what that enzyme does. So for example, lipases, it's an enzyme that breaks down lipids. That's an enzyme that breaks down proteins. That's an enzyme that breaks down carbs, and these are enzymes that break down nucleic acids. And so they really did try to name these nice and easy. That's going to get more complicated though as we go through. Okay, to understand where we're going next, you have to understand organic molecules. And Everybody has basically pushed all of this out of their memory from last semester when we talked about this in Biology 1, so I wanted to give you just a little refresher on your organic molecules. So this includes carbohydrates, proteins, lipids, and nucleic acids, the 4 things that we mentioned that were enzymes to help you digest just a little bit ago. In the world of carbohydrates, they can either be simple or they can be complex. Simple essentially is going to mean small. And small is going to be absorbable. If you can absorb it, you can put it into your bloodstream and you can use it as a nutrient. If it is bigger than that, it's complex, and that means you're either going to have to be able to digest it down until it's small, or it's going to pass directly through your system and you're not going to be able to use it. So, to give you some words that we used a little bit last semester, starch is a polysaccharide. Plants use it to store glucose inside of their cells, so if they're really good at photosynthesis, they make a lot of starch. Disaccharides are not quite as big. They are just 2 simple sugars that are put together, and that includes stuff like sucrose, which is a glucose and a fructose bonded together, or lactose, which is a galactose and a glucose bonded together. And we're not going to talk too much about oligosaccharides. They're branched chains of carbohydrates, but if you can digest starch down to a disaccharide, then you need a specific enzyme to break down the disaccharide, because these are also too big and you can't absorb these as is. If you had me for the first semester of this course, I spend a fair amount of time talking to you about lactose intolerance, and so we already mentioned this stuff last semester. If you don't have the enzyme, you can't break it down, it passes through your system, and then the bacteria in you digest it because you couldn't. Once you can get things down to a monosaccharide, which is these guys down here, that's a simple sugar that is small enough that you can actually absorb it. So you have to have a bunch of different enzymes to help you break down complex carbohydrates, because every enzyme is specific and it can only do one thing, and you've got a bunch of different kinds of sugar that are available to you from the food that you eat. So there's a table towards the bottom of page 181. There's a couple of mistakes in that table that we need to talk about. I'll get to those at the very bottom when I get to the lactase stuff. But in your mouth, you have several different sets of salivary glands. They produce saliva, and part of saliva is salivary amylase, which helps you break down starch. So if you put something like a cracker in your mouth, you can start breaking down the starch in that cracker in your mouth with chemicals like salivary amylase. Now, here's a weird thought for you. Enzymes are proteins, and proteins are also nutrients that you can consume. The salivary amylase that you make in your mouth, when it gets to your stomach, you digest it and you use your own enzyme as a fuel source to power the cells that are in your stomach, which means you're no longer digesting starch anymore because you just ate the enzyme that ate the starch. So you have to produce a whole new enzyme after you get out of the stomach, and that is pancreatic amylase. It does the exact same thing salivary amylase does, but you had to make a new one because you ate the first enzyme. After that, once we have broken down starch into maltose, we have to break down maltose. There are other sugars that are available in the foods you eat, like if you eat sweet tea, drink sweet tea, that has sucrose in it. So there's sucrase that can break down sucrose. There's maltase that can break down maltose. And then we get to the part that needs a correction. The table, 14.1 at the bottom, it says that the enzyme is lactose, and it is not. The enzyme is lactase, and so you need to make sure you change that in the table. Lactase breaks down the substrate lactose. It is produced by the small intestine to break down the sugar in the milk and cheese that you consume. The end products are also wrong in the table at the bottom of page 181. It says that lactose is broken down to glucose and fructose, but it's not. It's broken down to glucose and galactose. There is no fructose in lactose. So you need to make sure you change those two things. The enzyme is this, and the end products are that. Everything else in that line is okay. All right. Proteins. They are big, complex molecules made out of amino acids. Most have hundreds, if not thousands, of amino acids. If you remember in Biology 1, we talked about proteins having to have a very specific shape. They get twisted and folded into primary, secondary, tertiary, and sometimes even quaternary structure. And if that shape doesn't work, we talked a little bit about denaturing. Well, here, that's going to matter to us because the names change depending on whether we're folded or not folded. So if I say protein, I mean made of amino acids, and it's folded and it has a function. a polypeptide is still made out of amino acids, but it's not folded. It doesn't have the primary, it still has the primary, but it doesn't have the secondary, tertiary, or quaternary structures anymore. It only has the primary structure. So really, a polypeptide is just a string of amino acids. The amino acids are the individual building blocks of a protein. We talked about this not only in organic molecules, but we also had a whole chapter on protein synthesis in both lecture and lab. And so proteins should be something that are sort of in your head from last semester. So this is just trying to show you how we end up breaking those things down. This is what we eat in our food. I'm going to have some arroz con pollo tonight for dinner. So the chicken in that's got a lot of protein in it. I have to break it down into large polypeptides and then smaller polypeptides, and eventually I'll get it down into the amino acids that make up that chicken, and then I'll be able to absorb it. You can't absorb protein, it's too big. You can't absorb these guys either because they're too big. Only the amino acids are small enough for you to actually be able to absorb them. The enzymes that help you do that, you have several again. In your stomach, you have pepsin. It basically just unfolds the protein. Once we unfold it, we send it to the small intestine. The pancreas dumps some trypsin and chymotrypsin into the small intestine to break it down into smaller polypeptides. And sometimes we can also break it down into amino acids, although really we're just going to be breaking it down into smaller polypeptides there. And then protease is where we're actually going to get down to the amino acid level, and protease is made by and used in the small intestine. Lipids are complicated for several different reasons. One, they don't do the monomer-polymer thing that carbs and proteins do. Like, they're not made out of smaller building blocks. A lipid's just a lipid. Two, lipids are complicated by the fact that they do not mix into water. And your digestive system is water-based, which means the lipid likes to just sit there and float on the surface. And your enzymes can't get to it if it's floating on the surface of the mixture. And so we're going to have to do something called emulsification. Emulsification is how we mix lipids into water because it won't do that on its own. Probably at some point you've done something like made yourself a weird little wave lamp or wave bottle where you put some olive oil or something on the top and then you've put some water in it. And you'll notice the olive oil and the water don't mix together. That's what I'm talking about. If you were to put an emulsifier in there though, the olive oil would mix with the water. That's what emulsification actually means. So there's a couple of different chemicals that are going to help us work here. The liver is going to make bile. That's what carries out the emulsification process. Once we're emulsified, then we can actually start releasing enzymes, and lipases are enzymes that break down lipids. Lipids are mostly going to be triglycerides. This whole entire thing is one triglyceride. What we can do is break the bonds right here so that we can release a glycerol and then 3 fatty acids when we digest a lipid down. All right. Next, there are some test reagents that if we were going to be meeting face-to-face, we would have used. You're just going to have to watch me show you pictures of what it would have looked like. But these reagents help us test for certain chemicals to see whether or not digestion happened. Benedict solution changes colors when it mixes with simple sugars like glucose or fructose. Iodine turns, I say black, but some people say it looks more purpley or dark blue when it mixes together with starch, which is a complex carbohydrate. And then the other one we're going to use is blue litmus, and probably at some point in high school you use litmus paper. It is a pH detector. We are using a liquid form of it though, but it still works the same way, so it turns pink or red if it mixes in with acids. Now, another thing that's going to matter, if we were going to be mixing these tubes together, one of the chemicals you would be playing with is one called pancreatin. Pancreatin is a solution that is produced by the pancreas, and it contains all of the enzymes that the pancreas makes. And so I've gone ahead and listed those out, but just to say them again, pancreatic amylase digests carbohydrates like starch, trypsin and chymotrypsin digest proteins, and then lipase digests lipids. And so that pancreatin contains enzymes that can break down every major category of nutrient that you consume during any meal. All right, so page 183, enzyme experiments, digestion of starch. If we were meeting together in person, you guys would mix these tubes up, but we're not, so I'm just going to show you pictures of what things would look like so you would see how it was going to go. But here's what we're going to have. So test tube A, you would have mixed starch together with that pancreatin, which contains the enzymes. In test tube P, you would have put starch and then just some water so that they both had the same volume in them. What color are the tubes is a question under that little green chart. This is what color they would have looked like. And so it's cloudy or it's opaque. It's kind of whitish, but it doesn't really have a super pronounced color. After you mix these together, you would put them in the water bath, and then we would have to wait for the digestion to occur. But there's another question that it asks that we would have been talking about while we had the stuff in the water bath, and that is, which of these two tubes do you think would show evidence of digestion? Test tube A, that had starch and pancreatin, or B, that just had starch? Hopefully, if you think about it, you would think it was going to be test tube A, because that's the one that has the pancreatin, which has the enzymes. You have to have enzymes to do chemical digestion. There are no enzymes in test tube B, so that means there can't be digestion in test tube B. All right. After an hour, you were going to retrieve your tubes. This is what your tubes would have looked like, assuming you would have mixed everything together like you were supposed to. We would mix some Benedict solution in with that, and that would change the colors. Benedict solution is blue, naturally. After we put the Benedicts in, we would heat both of these two tubes. So they have both been heated and they have both had the Benedict solution added to them. So what color are the tubes before we heat them? And that is, they would both be blue. So you're filling in the blank. What color are tubes A and B currently? Blue. Then put both test tubes in boiling water for 3 to 5 minutes. And then this is the color they would come out as. Now here's where I want you, we're going to skip ahead for just a second, flip to page 184. And it says color after boiling. This is test tube A. What color is that after boiling? I'll take anything like green or orange or yellow. It's sort of a greeny orange, but whatever it looks like to you, I'm perfectly happy with. Is it positive for sugar? Anytime we get a color change like this, it's going to be positive. And so, yes, this is positive for sugar. The explanation for that. So, we did not put sugar in test tube A. We put starch in test tube A. Benedict's doesn't test for starch, it tests for sugar. So, how did sugar get in the tube if we didn't put sugar in the tube? Well, the pancreatin digested the starch, and when we digest starch, we release sugars. And so that's why we got a positive result. We digested starch down to sugar. In that same test tube, in that same test tube rack, test tube B, color after boiling, it's blue. Is that positive or negative? It's negative. Explanation, starch is still in there, and there's no sugar because there was no enzyme to break it down. All right, now, flip back ahead to page 183. And it says, I was going to mix tubes C, D, and E for you, and I've gone ahead and put what is in each one of these. All of these tubes are going to represent controls, except for A and B that are pictured up here. But C, D, and E, they're all controls. So maltose is a simple sugar. Water's water. Starch is a complex sugar. These are going to help us get positive and negative controls in future tests. These tubes that I'm showing you, we've already added the Benedicts, and we have already put them in the hot bath. And so flip back over to page 184, and test tube C. What color is it after boiling? It's orangey red, so that's this tube right here. And is that positive for sugar? Yes, it is positive sugar. Now, the explanation, we put sugar in that tube. It should have been positive for sugar. That is what's known as a positive control, and that's going to take me to the next slide. Anytime you're doing any sort of chemical experiment where you're using reagents, you want to have a positive control and a negative control. A positive control shows you what a positive test is supposed to look like, and it makes sure that your reagent works like you're supposed to, your chemicals were mixed like they were supposed to, and that everything is working like it's supposed to. So the fact that test tube C changed colors means everything was working correctly. We can trust the results on test tube A that had also given us positive controls. Negative controls show us what a negative test would look like. Test tube D, I don't have a picture for, unfortunately, but test tube D only had water in it. Since it only had water in it, it doesn't have starch, it doesn't have sugar, it would be a negative test. And so in the blank on page 184 for test tube D, the color after boiling would be blue. It is negative for sugar. And the explanation is it's the negative control because we didn't put any starch or sugar in there. There should not be sugar in there. So it's making sure that our negatives are all going to give us viable results. Again, what that means is we can trust this result because our negative control works like it was supposed to. So that also helps you just get your explanation because you can see what positive and negative tests are supposed to look like. Just to kind of prove my point or come back to these controls, anytime you go for like a COVID test, since that's something that's happening a lot right now, those tests have to have positive and negative controls too, just to make sure the test works like it's supposed to. So that same sort of thing is happening within that test strip. If it's a rapid test or within the PCR test, they always run it with a positive and negative control to make sure everything is working right. Now those controls are actually better on the PCR test, which is why the PCR test, which is the horrible nasal swab that they stick like all the way back to, it feels like your brain when you're getting them. But those tests are more reliable. They give less false positives and false negatives than the rapid test, which doesn't have as good a control feature that the PCR test happens to have. Okay, starch. Page 184, number 2, testing for starch. If you were in class, you would add some iodine to test tubes A and B. I would do the same thing for test tubes C, D, and E. Now, what iodine does is, if it contacts starch, it turns a very dark color. Again, to me, it looks black. Some people think it looks purpley or something, though. For test tube A and B and C, I don't have pictures of what those would look like. A would be positive. It would be black. Positive for starch. And the explanation for that is we put starch in the tube. And yeah, there was pancreatin in there, but the enzymes never digest all of a nutrient. There's always going to be some of it left. And so the reason it turns black is because there's still some starch left in the tube even after digestion has occurred. Test tube B. Let me remember what went into test tube B. Starch and water. That is going to give you a positive result. So test tube B would be black. Positive, and the explanation is we put starch and water in that tube. It should have had starch in there. Test tube C would also be black. Give you, wait, let me see what we put in test tube C. Sorry, I might be screwing up. Sorry, it's a negative test. It would be amber. Amber tests are the negative tests for iodine. And now I'm wondering if I said that wrong for test tube. No, yeah, B was positive still. So I think we're good. I just screwed up C as I said that. So C would give you the amber color that you see right here. And that's because we never put any starch in that tube. And maltose doesn't spontaneously make starch when you mix it into a test tube. Test tube D, amber. It's negative. And that's because that tube only had water in it. Test tube E, it only had starch in it, and you can see it turns black. That's positive for starch, which is exactly what it was supposed to have. Okay. Next up, lipids. This is one where we're going to be using the creamer if we were in the blue litmus. If we were together, you would be mixing these tubes for yourself, but of course we're not. So in one test tube, we would mix together half and half, which is cream like you could put in your coffee. Blue litmus, which is our pH indicator, and then pancreatin, which has our enzymes, one of which can digest fat. In the last test tube, we would put the cream, which has fat in it, the blue litmus, and then just water, so there's no enzymes in test tube G. It says mix and observe the color. Both of the tubes would be a light blue, and then we would put it in the water bath for an hour, and this is what it would look like after the hour. So let's see. Retrieve test tubes F and G from the water bath. This is towards the bottom of page 185. And it says, what is the color of test tube F after 1 hour? So F is this tube. It has become a little bit more pink. So is that positive for fat digestion? Yes, because blue litmus turns pinky red sort of in an acidic environment. The explanation for that is the enzymes digested the fat, released fatty acids, and that dropped the pH in that tube. Test tube G, color after 1 hour. It's the same blue that it was when we first put it in there. That is negative for fat digestion, and that's because we never put any enzymes in that tube. Okay. Top of page 186, demonstration of digestion of protein. So what they do for this one is they take a couple of hard-boiled eggs. They put one into a beaker of just water, And then they put one into a beaker of pancreatin. And remember, the pancreatin has enzymes that can digest everything, protein included. Now these I usually pass around so you can see them, and it's a little bit harder to see this when it's in picture form, but no matter how long this egg has been sitting here in this water, even if it's been a week, you can still 100% tell that's an egg. It has a smooth surface. It doesn't look like it's been altered at all. This one, on the other hand, the egg has dissolved, or to use appropriate terms, it's been chemically digested, and there's just a sediment of goo at the bottom of this, and that's because the pancreatin has digested the egg in the second one, and you can't, there's nothing in there. Like, you wouldn't be able to move it around to see an egg, because that powder at the bottom is the egg, so that's what it does to protein, it digests it. So for your observations, in the 5% pancreatin, you see a sediment of digested egg. And yeah, that's evidence for digestion. And then in I, you see a solid egg. Is there evidence of protein digestion? No, because you still see the whole egg. After that, we get to questions. Not going to talk through these a whole bunch because I've already said almost all of this. So once again, just pause the video and get what you need for the answers. But the picture on page 186 goes with the top table on page 187. So this is what you should fill into that table. Complete the following chart. This is what would complete that chart for you. And then questions 3, 4, 5, and 6 towards the bottom. There's the answers to those. Once again, pause the video to get your answers. Slew of questions on page 188. There's the first 4 answers. And there's all the remaining answers. Okay, page 189 starts the critical thinking questions, and for these I will slow down just a little bit for you. Number 1, a friend wants to lose some weight. What nutritional advice would you give them? Well, first off, you guys should not be giving anybody nutritional advice because you don't have a degree in nutrition and you haven't passed any certifications or bars in order to give anybody advice, so don't do it. But the sensible advice to give a person is you still have to eat a balanced diet. Nobody should ever eat fewer than 1,200 calories per day, because if you do, it's a guarantee that you are not going to be getting all of the vitamins and minerals you need, which means you're going to start to develop deficiency symptoms. And probably at some point in your life you've heard of something like scurvy or osteoporosis. Those all result from a lack of a good diet. And so don't ever go below 1,200 calories per day. You do, however, want to reduce the number of calories you're consuming if you're trying to lose weight. You just have to do it sensibly. You need to eat more dark leafy greens like spinach and kale and collard greens. You need to eat less sugar, especially. Sugar is crazy addictive and it's really hard to beat, but sugar flat out makes you fatter, and so you've got to cut the sugar out of your diet. And if you're really trying to lose weight, you got to add exercise in there. And like I say, there is no shortcut. There's no magic pill you can take, period. You have to do the work if you want to lose weight. Number 2, a second friend is contemplating gastric bypass surgery. What specific parts of the digestive system would be affected? The full Ruin Y gastric bypass surgery, or Ruin X, I don't remember. But anyway, that particular procedure removes most of the stomach and a big long chunk of the small intestine as well. By doing that, it impacts you 2 ways. 1, you cannot eat a big meal after you have a gastric bypass because your stomach is now about the size of a quarter. If you try to eat a big meal, guaranteed you're going to puke. I had a student several years ago whose mother had the procedure, and he said that his mother could not even eat a full Go-Gurt tube. Do you guys remember those tubes of yogurt like your mom would put in your lunch at school or whatever? She couldn't even eat one of those tubes without vomiting. She could only eat part of the tube. So yeah, you can't eat a lot at one time. The other way that it affects you is if you've taken out most of your small intestine, that means you've taken out most of the organ that absorbs the nutrients for you. So you're not going to be absorbing things very well. Now, while those are the only organs that are removed during gastric bypass, this is going to have major impacts on all the other organs of your body. Your liver is going to be affected, your pancreas is going to be affected, your large intestine is going to be affected, the feces that you produce is going to be very different than it was prior to having the surgery. It just impacts everything. a true gastric bypass, not like a sleeve or a lap band or something, you have to go in for psychological evaluation before you get it done, because it is no joke, that procedure. It almost guarantees nutritional deficiencies in the future, and so it's scary, that particular procedure. Number 3, if a person has their gallbladder removed, how would that affect their digestive system, and how would that change, or how should they change their eating habits? Well, remember, your gallbladder's job is to store bile and release it after you eat a fatty meal. If you don't have a gallbladder, your liver does still make bile, but it can't release a bunch at once like you would need to do if you ate a high-fat meal. As a result, you cannot emulsify the fat like you're supposed to, so the fat, again, just floats on the surface of everything and you never digest it. Now, here's where I'm going to get gross for just a second. Any of you that are old like me, you might remember there was a chip that was made ages ago called, what they call the chip? They fried it in something called olestra. Olestra is a fat that human beings are not capable of digesting or absorbing. And unfortunately, they had to put a warning on the bag of chips. And the warning is the reason why that bag of chips doesn't exist anymore. And it was, eating these may cause anal leakage. Your anal sphincter is not very well adapted for keeping a bolus of fat inside of you. So if you don't digest the fat, it just passes through your system as basically a little ball of fat, and it just slips right through your anal sphincter. And so basically you sort of poo droplets of fat all day. And that's not a very pleasant thing for anybody to have to go through. And so that's essentially a fatty diarrhea that ends up developing, but it will only develop if the person doesn't watch what they eat. So kind of coming back to this question, how should they change their eating habits? Do not eat a meal that is high in fat. You will full on regret it if you choose to do that. You can still have little bits of fat, but you can't eat a bunch at one time because you can't release a bunch of bile at one time. So you just have to moderate how much fat you eat. Now, there is also an implication for some vitamins. Some vitamins are fat soluble, and so some people who have their gallbladder removed develop deficiencies in those fat soluble vitamins. They will end up being put on supplements to help them try to get the vitamins that they need. Next, what are ulcers and how are they treated? Ulcers are essentially little holes that you digest, usually in the stomach. Your stomach acid literally starts to eat your stomach when it's not supposed to. Not that it ever is supposed to. This causes bleeding, and so a person will very often start to vomit up blood, or if they don't vomit it up, it ends up turning the stool or the feces very, very dark black because of the digested blood that is in the feces. It's very painful when a person has ulcers, and so they usually will go in complaining of heartburn or a very, very sharp, I don't know how else to describe it. It's like somebody stabbing you in the stomach when you have an ulcer, and the stabbing pain just doesn't go away, essentially. The old school treatment for this was just antacids and watch what you eat, like don't eat spicy foods or don't eat triggers. But it has been shown, actually in the 1990s, it was shown that ulcers develop as a result of a Helicobacter pylori infection. That's a species of bacteria. So now antibiotics are usually used to treat them. Number 5, what are vitamins and minerals? So vitamins are organic molecules. Minerals are straight up elements you can find on the periodic table, like calcium, iron, nickel, copper, those sorts of things. What they do is they help enzymes function in your body. And remember, enzymes help you carry out every single metabolic process that you need to be able to carry out. What that means is your vitamins and minerals help you carry out those processes. So if you have a crappy diet and you're deficient in a vitamin or a mineral, that means there's an enzyme that can't do its job because the vitamin isn't there. And that means there's something in your body that you can't do as a result of that not being there. So I'll give you an example. The one vitamin deficiency that most people have heard of is scurvy. Scurvy results from a lack of vitamin C. And incidentally, cases of that in the United States are on the rise, especially among college students, because they don't know how to eat a good diet. If you go with the traditional pizza, ramen noodles, macaroni and cheese, I'm trying to think of what else is like a staple food that college students tend to rely on, fast food in a lot of cases too. Those things don't have the vitamin C that you need, and so scurvy is on the rise. If you don't have vitamin C, you can't heal wounds, and your connective tissue sort of starts to dissolve. And so what that means is if you do develop a wound, it's not going to heal. It's just going to stay there. That includes like a blister on your foot. It's just never going to go away. It also means your teeth are going to tend to fall out of your mouth because connective tissue is what holds your teeth into your bones. And so without that connective tissue, your teeth fall out. So you've got to make sure you're eating, again, a well-balanced diet so that you're getting not just the carbs, proteins, and lipids that you need, but also all the riboflavin that you need and all the folic acid that you need. Vitamins and minerals are critically important, even though you only need a teeny tiny amount. The other thing that I did want to say is my PS there. Just because you need them does not mean you need too much of them. I have heard people say in relation to vitamins and minerals that if a little is good, a lot must be better. And that is not true. You can overdose on vitamins and minerals, especially the minerals. In fact, one of the leading causes of overdose in children is iron, because they will eat too many of their, well, it used to be Flintstones vitamins. I don't know if they still make those or not, but those vitamins do have iron in them, and too much iron will flat out kill you. It will make your liver die, and then you will die as a result. And so you don't want to get an excess of those vitamins. Now, that's easier to do, get an excess of your fat-soluble vitamins than it is your water-soluble vitamins, but you never want to get 3,000% of something during the course of a day. So my other little side note to make as related to that is, if you're a person who drinks a lot of energy drinks, check those food labels, guarantee you're overdosing on something. And so watch out for those. Number 6, how do fungi eat? That question is so out of left field, but they are decomposers, but they don't have mouths. They can't ingest. Instead, what they do is they release their enzymes outside of their body to break down the food in the soil or water or wherever they happen to be living, but usually soil. After they have broken it down, then they can absorb it into their body. So they do the digestion outside and then absorb. We kind of do it a little bit differently in that we ingest our food first. And so that completes the digestive system chapter. Make sure you take your digestive system post-lab quiz.